Hi, I'm Jose Clemente. I'm an assistant professor at the Department of Genetics and Genomic Sciences at Mount Sinai in New York City. Today, I'd like to share some results with all of you that we have recently published in the journal Science Advances with colleagues from NYU, WashU, UCSD here in the US, with the CAICET, IVIC, and Instituto de Medicina Tropical in Venezuela, and with University of Alberta in Canada. There are a number of lifestyle practices that are dramatically different between 100 years ago and now. We did not used to have antibiotics. We did not have C-section. Our diets were completely different. And all these factors, antibiotics, C-section, diet, they can reshape the type of bacteria that we have in our bodies in a very significant manner. The question that we had was, what was the human microbiome like before we incorporated all these practices into our lifestyle? And I think this is an important question, because if we can figure out what we used to have, what bacteria we used to harbor in our bodies, and which bacteria we have lost, we might be able to gain very valuable insights into mechanisms of pathogenesis. Why are these diseases now so common? Is it related to the fact that we're losing bacteria that were exerting a protective effect on the host? We were extremely fortunate to collaborate at the time with Maria Gloria Dominguez, a colleague and good friend of ours from New York University. She had access to a collection of samples from a Yanomami village that was first contact in 2009. This village is located in the Alto Orinoco State in Venezuela, close to the border with Brazil. So 11 of the villagers gave consent to be sampled. And we obtained samples from the skin, samples from their oral mucosa, and stool samples to get an idea of what the microbiome was in each of these body sites for the people who were part of this study. And then what we did was we sequenced these samples. We got an idea of what bacteria this particular village has, and we compared that against 3 other populations. We compared it against the microbiome of Guahibo Amerindians, also from Venezuela, in a different location. We compared it against Africans in 4 rural communities in Malawi. And then we compared it against a group of US subjects. What we found was that these Yanomami Amerindians, they had unprecedented levels of bacterial diversity. And of course, they have much more diversity than we have. This is to be expected, of course. But they also have significantly higher levels of bacterial diversity than the Guahibo Amerindians or the Malawians. And this is important because both the Guahibos and the Malawians, they still live very traditional lifestyles, but they have been already impacted to some extent. They use antibiotics, they occasionally consume processed foods. So what this is suggesting to us is that perhaps even minimal exposure to any of these practices can have a significant impact in the amounts of bacterial diversity that we have. So the next step for us are to figure out more precisely what functions these bacteria that we are losing are playing in the host, to figure out how those functions the absence of these functions might be important in the pathogenesis of several of these immune and allergic diseases that we're discussing, and to figure out how we can best restore these functions that we are losing but that we used to have. To find out more about our study, you can read our paper in Science Advances, or you can check our website.